Welcome to your quiet corner of cosmic discovery. I'm here to share something genuinely fascinating about our universe at just the right volume for drifting off. You know what's fascinating? I've been thinking about how our bodies are like tiny battlefields, quietly defending us all day and night without us even noticing. There's something remarkable about how the immune system can tell friend from foe and win those microscopic battles, and I can't wait to share the amazing ways it keeps us safe, even when it seems like it's fighting itself. Should these dreamy explorations accompany your journey to sleep, a gentle like or subscription would be deeply appreciated. What questions about our universe keep you wonderfully curious? Let's prepare for tonight's journey. Find your most comfortable position, let tension float away from your body, and take 3 slow, peaceful breaths. There are stories that unfold on distant planets, in the heart of nebulae, across cosmic wastelands lit by the shimmer of 1,000 unfamiliar suns. Science fiction has, for generations, taught us to imagine conflict on a grand scale. The technologically advanced facing the unknown, the familiar pitted against the alien. Yet, of all the battlefields dreamed in books and on screen, perhaps the most profound is one that lies unseen within each of us. As you lie back and let your thoughts drift, consider for a moment the silent, ceaseless struggle taking place beneath the surface of your skin, in the labyrinthine corridors of tissue and fluid that make up your body. This is the story of the immune system, a relentless, unending saga of vigilance, defense, and adaptation. Long before H.G. Wells penned his prescient lines in The War of the Worlds, the drama of survival played out at scales invisible to the naked eye. The Martians in Wells' tale fell not to the guns or ingenuity of humankind, but to bacteria, microbes that inhabit our world and indeed our bodies. Wells, in his quiet way, pointed to a truth that biologists would later fully appreciate. The smallest actors can wield the greatest power. The defeat of the Martian invaders was not a sudden deus ex machina, but the inevitable verdict of an evolutionary arms race that has been running for billions of years. Consider the vision of a body as a fortress, or perhaps more accurately, a vast living city. Its walls are not stone, but skin and mucous membranes, the first lines of defense, patrolled by sentinels that scrutinize every visitor. Within, a network of highways and alleyways, blood vessels, lymphatic channels, carry not only nutrients and messages, but also the agents of defense, ever alert for signs of intrusion. Just as in the Starship Enterprise from Star Trek, where a crew works in concert to face threats both internal and external, so too do our cells and molecules operate in harmonious complexity, each with a role to play in the maintenance of the whole. The immune system is both a shield and a sword. It is decentralized, distributed throughout the body, yet it acts with remarkable coordination. This is not a monolithic army, but a coalition of specialized forces, each trained for a particular kind of threat. There are border guards, wary of anything that does not bear the right credentials. There are rapid response units, ready to flood a site of injury or infection with overwhelming force. There are intelligence analysts cataloging past encounters and adjusting future strategies accordingly. All this takes place without conscious input, day and night, from the moment we are born until our last breath. Imagine now the first moments of an invasion. A splinter of wood pierces the skin, and with it a handful of bacteria, eager to carve out a niche in this new world. For them, your body is a universe, an expanse of resources and possibilities. They are the would-be colonizers, the Martians of Wells' imagination seeking to bypass the barriers that keep the world at bay, yet they are not unopposed. Within seconds, the local patrols, cells called macrophages, have detected the breach. Their name comes from the Greek for big eaters, and these are the oldest and most tireless of our defenders. They engulf invaders whole, digesting them in a chemical tempest, and in doing so send out alarms, summoning reinforcements from distant regions. This is the innate immune system, ancient, broad spectrum, and always ready. It is the equivalent of the starship's shields, raised at the first sign of danger, indiscriminately repelling all comers. Macrophages, neutrophils, dendritic cells, each is a player in this initial skirmish. Yet the innate system is only the beginning. If the invaders persist, if they are clever or numerous or simply lucky, the battle escalates to a new phase, one that is as intricate as it is deadly. Here we find the adaptive immune system, perhaps the most sophisticated defense mechanism evolution has ever produced. Where the innate system is blunt and immediate, the adaptive is precise and calculating. It learns from every encounter, remembers each foe, and tailors its response with exquisite specificity. Like the tactical teams on the Enterprise, it can analyze the enemy, devise a new strategy, and deploy specialized weapons manufactured to fit the threat precisely. Lymphocytes, T cells and B cells, are the architects of this response, each bearing unique receptors forged through a process of genetic shuffling, a molecular lottery that ensures our immune repertoire is as vast as the diversity of pathogens we might ever encounter. Here, science fiction and biology intertwine again. In Star Trek, when the crew faces a novel organism or an unpredictable adversary, they do not simply blast away with phasers. They study, adapt, and reconfigure. The immune system operates in much the same way. B cells, upon recognizing an invader, may transform into antibody factories, flooding the bloodstream with proteins that lock onto the pathogen, marking it for destruction or neutralizing its abilities. T cells, meanwhile, may directly kill infected cells or orchestrate the broader response, coordinating the activities of other immune agents. The scale of this conflict is staggering. In every drop of blood, thousands of immune cells circulate. In the marrow of your bones, new recruits are constantly trained and released, their fates determined by signals from their environment, and a relentless testing process that ensures they will not turn against the body itself. The lymph nodes, those small, bean-shaped way stations scattered throughout your tissues, serve as strategic command centers where information is gathered, threats assessed, and orders disseminated. Yet, despite its power and precision, the immune system is not infallible. Like any complex defense, it is vulnerable to subterfuge and sabotage. Some viruses, such as HIV, are masters of disguise, cloaking themselves from detection or attacking the very cells meant to destroy them. Others, like the influenza virus, mutate rapidly, changing their appearance with every generation, forcing the immune system into a perpetual game of catch-up. Here, too, the analogies with science fiction abound. The shapeshifters and chameleons of alien lore, always one step ahead of the hunter. But it is not only pathogens that threaten the integrity of the inner battlefield. The immune system, in its zeal, can turn upon the self, mistaking the body's own tissues for foreign invaders. Autoimmune diseases, lupus, rheumatoid arthritis, type 1 diabetes, are the tragic result of friendly fire, the consequence of a defense that cannot always distinguish friend from foe. Like a starship's computer misidentifying its own crew as intruders, these errors are costly, often devastating, and remind us of the delicate balance that must be maintained at all times. Throughout all this, the immune system is not a static entity. It evolves within a lifetime, shaped by experience and memory. Vaccines are one of humanity's most successful attempts to harness this capacity, training the immune system with harmless mimics of dangerous pathogens, so that when the real enemy appears, the response is swift and overwhelming. Here, the analogy to science fiction becomes almost literal. We are, in a sense, programming our defenses, uploading new protocols into our biological software. Consider again the Martians of Wells's tale. Their defeat was not a matter of chance, but the inevitable outcome of millions of years of coevolution between humans and the microbes of Earth. The immune system is the record of that history, inscribed not in books or archives, but in the very molecules and cells that patrol our bodies. It is an epic saga, written in blood and bone, of struggle and adaptation, of loss and triumph. And yet, for all its drama, the immune system operates in silence, beneath the threshold of sensation. You do not feel the macrophage engulfing a bacterium or the B-cell dividing furiously in response to an infection. You may sense the symptoms of war, a fever, a swelling, a fatigue, but the intricacies of the conflict are hidden from view. Like the cloaked Romulan ships in Star Trek, much of the action occurs out of sight, detectable only through its effects. To study the immune system, scientists have had to become detectives, piecing together clues from experiments, developing new tools to visualize the hidden world within. The electron microscope has revealed the bizarre landscapes of immune cells, bristling with receptors and granules, while molecular biology has allowed us to read the genetic instructions that underlie their function. Each discovery opens new vistas, suggesting that the battlefield is even more complex, the enemy more cunning, and the defenders more ingenious than we once imagined. And so, as you let your mind wander deeper into the twilight between wakefulness and sleep, picture your body not as a passive vessel, but as the setting for a story as rich and tumultuous as any recounted in fiction. The immune system is the unseen hand that preserves the fragile continuity of your existence, fending off dangers that lurk in every breath, every cut, every mouthful of food. It is the reason you can ponder the stars, dream of alien worlds, and contemplate your own place in the cosmos. The story of the immune system is, at its heart, a story of identity. It is the means by which the body distinguishes self from other, the mechanism that allows us to coexist with a multitude of microbes that inhabit our skin and gut, while defending against those that would do us harm. This is not a simple binary, but a dynamic negotiation, a perpetual balancing act between tolerance and aggression, between openness and vigilance. Perhaps, in the quiet of the night, there is comfort to be found in the knowledge that, even as you sleep, this inner war is waged on your behalf. The soldiers never tire, the sentinels never rest. They are the legacy of countless generations, the product of an evolutionary struggle that predates all human history. They are the reason that life, in all its vulnerability and promise, can persist against overwhelming odds. But the story is far from over, for even as the immune system defends and remembers, it must also learn. Each encounter with a pathogen is a lesson, each victory or defeat a chapter in an ongoing saga. In the chapters to come, we will delve deeper into the mechanisms of this learning, the ways in which the body records its experiences and adapts to new threats. For now, let the image linger. A city in the night, lit from within, its defenders vigilant, its boundaries secure, for the moment, against the endless tide of the unknown. Beneath the gentle surface of your skin, far removed from daylight and the comforting regularities of conscious life, there unfolds an endless game of exquisite strategy, a contest as old as multicellular existence itself. This is the immune system, not a single organ nor a simple wall of defense, but a living network of alliances and surprises, a chessboard drawn in the shifting patterns of blood and tissue. Here, every piece, each cell, molecule, and signal moves according to rules both ancient and evolving, navigating a balance between vigilance and restraint, collaboration and solitary action. In this twilight, where the self is constantly defined and defended, even the most familiar cells might one day become a stranger, and every new visitor is subject to scrutiny. To step into this realm is to slip through a hidden door to witness a city in perpetual motion, The sentinels, the messengers, the assassins, and the healers, each has a role, and each must play it with precision, for a single misstep can tip the balance from health to chaos. Yet, as with any grand game, what appears at first to be orderly can, on closer inspection, reveal layers of unpredictability, miscommunication, and unintended consequence. The opening moves belong to the innate immune system, humanity's ancestral shield. Its guardians are ancient, their tactics blunt but effective, honed over evolutionary time. Neutrophils, the most abundant of the white blood cells, patrol the bloodstream in restless search, their lifespan measured in brief, furious days. When the skin is breached, or when a virus dares to slip past the mucosal barricades, these cells are among the first to arrive. They engulf invaders in a molecular embrace, unleashing toxic granules and reactive oxygen to dissolve their prey. In their wake, they leave a battlefield strewn with cellular debris, the first sign to the wider system that a challenge is afoot. Yet the neutrophil's sacrifice is rarely silent. Even as they perish, these cells release signals, tiny chemical flares that rouse other defenders. Macrophages, the great eaters, drift through tissues, ever hungry, ever vigilant. They devour the detritus left by neutrophils, clean up the chaos, and crucially present fragments of the enemy on their surfaces. Like chess players revealing a captured piece to warn their allies, macrophages display these molecular trophies, marking the beginning of a deeper, more nuanced game. The innate immune response is swift and general, a first line that cares little for subtlety. But it is not enough to simply react. Survival demands memory, adaptation, and foresight. This is where the adaptive immune system enters, a newer layer in the evolutionary palimpsest, as subtle and complex as any grandmaster's strategy. Lymphocytes are its principal players, divided into two great houses, B cells and T cells. Each is born with the potential for unique specificity, a molecular lock designed to fit just one kind of intruder's key. In the bone marrow, developing B cells undergo a process of recombination, shuffling the genetic deck to produce a vast diversity of antibodies, each a potential weapon against a different threat. The majority are discarded, deemed too dangerous or useless, but the survivors emerge ready to patrol the bloodstream, ever watchful. T cells, meanwhile, mature in the thymus, that small, mysterious gland nestled behind the sternum. Here they are tested not just for their capacity to recognize enemies, but for their loyalty to the self. Those that react too strongly to the body's own tissues are marked for destruction, a process that is imperfect but necessary. Some T cells are trained to kill directly, seeking out infected or cancerous cells with lethal precision. Others act as coordinators, releasing cytokines, chemical whispers that direct the flow of the immune response, summoning help or urging restraint. When a macrophage presents its captured fragments, it is the T cells that respond. If the pattern matches their own unique receptors, they are activated, proliferating into an army of clones. Some will seek out the invaders directly, others will rally B cells, prompting them to divide and secrete a flood of antibodies. These Y-shaped proteins bind to pathogens, marking them for destruction or neutralizing their ability to infect. In this way, the adaptive immune response is both personalized and amplified, capable of learning from each encounter and remembering past foes for years or even decades. But the immune system is not a well-oiled machine. It is a living, breathing chessboard with alliances that shift, strategies that sometimes fail, and pieces that may not always play by the rules. For every clear-cut battle against a foreign invader, there are moments of confusion and misrecognition. The immune system must constantly walk a tightrope. Too weak, and infection or cancer may flourish. Too strong, and it turns against the very body it is meant to protect. Consider the phenomenon of autoimmunity, a betrayal from within. Here, the immune system, in all its fervour, mistakes self for enemy. Cells meant to defend instead attack, laying siege to joints in rheumatoid arthritis, to insulin-producing cells in type 1 diabetes, or to the protective sheath around nerves in multiple sclerosis. The reasons for these betrayals are many and not fully understood. A mixture of genetic susceptibility, environmental triggers, and the random roll of cellular dice. Sometimes, a viral infection may so closely resemble a piece of the self that in its wake, the immune system loses its sense of distinction. Sometimes, the process of educating T cells in the thymus is incomplete, and a few rogue soldiers escape notice. Equally complex are the alliances, the moments when the immune system must decide not to act. Every day, the gut is exposed to an ocean of foreign material, bacteria, food particles, harmless antigens. Yet, for the most part, these are tolerated, thanks to a delicate truce negotiated by regulatory T cells and the microbiome itself. This is immune tolerance, a piece as fragile as any armistice. When it breaks, as in allergies or inflammatory bowel diseases, the result is a misdirected onslaught, a tempest against imagined foes. Within this living chessboard, communication is everything. Cytokines, tiny proteins secreted by immune cells, act as the signals, the shouts and whispers that coordinate attack, call for reinforcements, or declare a ceasefire. But this network of signals is not immune to error. Sometimes the response is too strong, as in the notorious cytokine storm, a runaway cascade that can damage organs and prove fatal, as seen in severe infections or certain reactions to new therapies. Other times, signals are lost or misread, leaving the body vulnerable or unprepared. There are, too, the shadowy games played by pathogens themselves. Viruses, bacteria, and even some cancers have evolved to manipulate the immune system, disguising themselves, subverting signals, or turning the body's own defenses to their advantage. HIV, for example, targets and destroys the very T helper cells meant to coordinate the immune response, leading eventually to immune collapse. Tuberculosis bacteria can lie dormant within macrophages for years, evading detection. Tumours may cloak themselves in molecules that mimic normal tissue or secrete their own suppressive signals, turning the immune system from hunter to unwitting accomplice. Such betrayals are reminders that the immune system is not a solitary actor, but a participant in a vast ecological contest, one where the lines between friend and foe are constantly redrawn. Even the commensal bacteria of the gut, once thought to be mere passengers, are now recognized as key players, shaping immune development, moderating inflammation, and teaching tolerance. These alliances are ancient and essential, yet always at risk of disruption by antibiotics, diet, or disease. If the immune system is a chessboard, it is one where the pieces are in constant flux, where the rules are written in code and the strategies adapt with every move. The notion of memory, so central to adaptive immunity, is itself a marvel of biological engineering. Some B and T cells, after a battle, become memory cells, lying in wait for a future encounter with the same foe. This is the principle behind vaccination, one of medicine's greatest triumphs. By presenting the body with a harmless version of a pathogen, we train the immune system to recognize and respond more rapidly when the real enemy appears. Yet even this is not foolproof. Some pathogens mutate so quickly that yesterday's memory may be obsolete by tomorrow. The immune system's complexity is further deepened by its relationship with time and development. In the womb, the immune system is carefully restrained to avoid attacking the developing fetus. At birth, it is naive, untested, relying on maternal antibodies passed through the placenta and later in breast milk. Through childhood, exposure to the world's microbial diversity shapes its repertoire, teaching it what to fight and what to ignore. In old age, the system falters, its memory cells dwindle, its responses grow sluggish or misdirected. The risk of infection rises, as does the likelihood of chronic inflammation, a slow-burning fire that can contribute to the diseases of aging. For all our knowledge, vast mysteries remain. The exact balance between immune activation and tolerance is still being mapped. The signals that distinguish friend from foe are not always clear. Autoimmune diseases, allergies, and immune deficiencies remind us that this system, for all its power, is fragile and fallible. We have learned to manipulate some of its parts through drugs that suppress or enhance, through therapies that redirect T cells to attack cancer, but each intervention is a calculated risk, a move on the chessboard that may have unforeseen consequences. And then there are the questions that touch on the very nature of identity. The immune system is, in a sense, the ultimate arbiter of self. It defines the boundaries of the body, decides what to permit and what to reject. Organ transplantation, for example, is not merely a surgical feat, but an immunological negotiation. Unless the recipient's immune system is suppressed, it will recognize the new organ as foreign and attack it. Yet, over time, some tolerance may develop, a grudging acceptance forged through careful management, and sometimes sheer biological serendipity. The immune system's reach extends beyond the physical. Emerging evidence suggests connections between immunity and the mind, between inflammation and mood, between the state of the gut and the workings of the brain. The chessboard is larger, more intricate than we ever imagined, its pieces moving not just in blood and lymph, but in the restless circuits of thought and emotion. In the quiet hours, as you drift towards sleep, consider the millions of decisions being made within you, cells whispering, signaling, moving into position, or standing down. The chessboard is never still, its pieces never truly rest. Each night, as you close your eyes, your immune system remains vigilant, balancing on the knife edge between protection and pathology, memory and forgetfulness, action and restraint. What strategies will unfold in the darkness? What alliances will be forged? What betrayals narrowly averted? The game continues, unseen but unceasing, as the body's silent sentinels patrol their endless domain, ever watchful, ever uncertain. As one move leads to the next, the mysteries deepen, drawing the mind onward into the labyrinth, where understanding is always partial, and the final checkmate forever just out of reach. The world of the immune system, so intricate, so fierce in its quiet vigilance, has not yielded its secrets easily. For centuries its workings remained hidden, an invisible ballet within the body, attended by neither the poet's gaze nor the scientist's lens. Yet, like any great mystery, its boundaries were slowly mapped by the steady hands of those who dared to ask, what lies beneath our skin, fighting on our behalf? To truly appreciate the landscape of the immune system, we must walk alongside these explorers, those who fashioned the first crude compasses and those who, of lifetimes, refined the tools and techniques that would illuminate the invisible war within. Let your mind drift back to the late 17th century, to the Dutch Republic, where a cloth merchant named Antonia van Leeuwenhoek peers curiously into a drop of pond water through a lens of his own making. He is not a scientist by trade, yet his fascination with the tiny world leads him to polish lenses with such precision that for the first time a human eye glimpses the teeming multitudes of life at the microscopic scale. Through his handcrafted microscopes, van Leeuwenhoek observes animalcules, what we now know as bacteria and protozoa, swimming like minuscule phantoms in a droplet. He does not yet know that these minuscule beings can sicken or even kill, nor does he imagine that the body itself possesses sentinels to stand guard against such threats. But in crafting his lenses, van Leeuwenhoek unwittingly forges the first tool of immunological discovery, a way to see the invisible. The microscope, in all its evolving forms, becomes the pioneer's lantern, shedding light on the landscape of blood and tissue. With it, later scientists would observe the cells that patrol our bodies, the white blood cells, squirming and shifting, their shapes as mutable as clouds. Across the centuries, the microscope's magnification improves, and with each leap, the boundaries of the immune system's battlefield become more distinct. In the 19th century, the German pathologist Paul Ehrlich, with his love for colour and curiosity, stains blood and tissue samples with aniline dyes, watching as different cells absorb distinct hues. Under his gaze, the immune system's foot soldiers, lymphocytes, neutrophils, eosinophils, acquire not only names but vivid identities, their forms and purposes gradually teased apart by the careful application of colour and light. But to see is not always to understand. The immune system is not a mere collection of parts, but a living, dynamic defense, and its motives cannot be read from shape alone. So the story of discovery moves from observation to intervention, from passive witness to active participant. Here, the narrative turns to the tools of manipulation, to experiments that do not simply watch, but prod and provoke, coaxing the immune system to reveal its rules. Consider the tale of Edward Jenner, a country doctor in 18th century England, who became both detective and healer. In an era haunted by the spectre of smallpox, a disease that ravaged communities and left its survivors pockmarked or blind, Jenner listens to the folklore of milkmaids who claim immunity to smallpox after suffering the far milder cowpox. His mind, alert to patterns, wonders. Could exposure to one disease shield against another? In 1796, Jenner scratches pus from a cowpox sore into the arm of a young boy, James Phipps, and later exposes him to smallpox. The boy does not sicken. Jenner's experiment, though crude by modern standards, reveals a profound truth, that the immune system remembers and that its memory can be trained. Thus the first vaccine is born, not from knowledge of cells and molecules, but from observation, inference, and a willingness to test the boundaries of nature's rules. The notion of vaccination, of training the immune system in advance, unlocks a new chapter of experimentation. The art of immunology, as the field would come to be known, flourishes in the hands of Louis Pasteur, who, with equal parts rigour and imagination, refines the process of attenuating microbes to create safer vaccines. Pasteur's work, and that of his contemporaries, relies on animal models, sheep and rabbits, chickens and mice, whose bodies serve as testing grounds for hypothesis and hope. It is through these living laboratories that the mechanisms of immunity begin to clarify, that exposure to a weakened or inactivated pathogen can grant protection against its virulent cousin. Yet, as the 19th century wanes, the immune system's workings remain only dimly understood. The tools of the time, microscope, dye, syringe, are blunt instruments, and the battlefield remains shrouded in fog. It is now that the story grows richer as rival theories contend and new methods emerge to settle the questions that observation alone cannot answer. One such question: does the body fight invaders with cells or with soluble factors? The debate is fierce, and the answer is won not by speculation but by ingenious experiment. In the 1880s, the Russian zoologist Ilya Mechnikov, observing starfish larvae under his microscope, notices strange cells engulfing foreign particles, devouring them whole. He theorizes with poetic audacity that these cells are the body's defenders, and that a similar process occurs in humans. To prove his point, Mechnikov inserts a rose thorn into the transparent larvae and watches as the cells swarm the injury, consuming the invader. He names these cells phagocytes, eating cells, and asserts that cellular immunity is the cornerstone of defense. Meanwhile, in Germany, Paul Ehrlich and Emil von Behring pursue a different line. They isolate the serum, the liquid fraction, of blood from animals immunized against diphtheria or tetanus, and show that it can neutralize the toxin in other animals. Their work reveals the presence of antibodies, soluble molecules that circulate through the blood, seeking out and binding to foreign invaders. The two camps, cellular and humoral immunity, debate, often heatedly, but in time it becomes clear that both are right. The immune system is a partnership of cells and molecules, each with its own role, its own strategies and tactics. As the 20th century dawns, the pace of discovery accelerates. New tools appear, each more precise than the last. The invention of the ultracentrifuge allows scientists to separate the components of blood by their densities, isolating plasma from cells, teasing apart the immune system's elements with ever finer resolution. Electrophoresis, a technique that uses electric fields to sort molecules by size and charge, reveals the diversity of antibodies, each a unique shape tailored to a specific threat. It is during these years that the first great maps of the immune system's terrain are drawn. The French immunologist Jules Bordet develops methods to measure the activity of antibodies and complement, another soluble factor critical to defense. In England, Rodney Porter and Gerald Edelman, working independently, use clever chemical tricks to disassemble antibodies into their component parts, revealing their Y-shaped structure and the secret of their specificity. For this, they are awarded the Nobel Prize, and the field of immunology is forever changed. But the immune system, ever elusive, guards its subtler secrets well. The tools of chemistry and physics alone cannot answer all questions. To understand how the immune system distinguishes self from non-self, how it avoids attacking its own tissues while recognizing the most fleeting trace of a pathogen, requires new forms of insight, new experimental designs. One such experiment unfolds in the 1940s in the laboratories of Peter Medawar and his colleagues. They wonder, can the immune system be tricked into accepting foreign tissue as its own? Working with mice, they graft skin from one strain onto another. In most cases, the graft is rejected, attacked by the recipient's immune cells. But when newborn mice are exposed to foreign cells, they later accept grafts from the same donor strain. Medawar concludes that the immune system learns what is self early in life, a concept that will echo through the decades in the study of tolerance and autoimmunity. To probe the immune system's responses at the most granular level, scientists turn to the tools of molecular biology. In the 1970s, the invention of monoclonal antibody technology by Georges Kohler and César Milstein marks a revolution. By fusing an antibody-producing cell with a cancer cell, they create immortal cell lines that churn out pure, identical antibodies, tools of exquisite precision capable of marking and tracking individual molecules on the surfaces of cells. With fluorescent dyes attached, these antibodies transform the microscope yet again, illuminating the immune system's actors in riotous color. The birth of flow cytometry soon follows, a technology that can sort and count thousands of cells per second, distinguishing lymphocytes from monocytes, T cells from B cells, by the unique constellation of molecules they display. The immune system's secrets, once the domain of guesswork and intuition, are now revealed in hues of red and green, blue and gold. The battlefield is charted not only by sight, but by sequence. The ability to read the genetic code of immune cells, to watch as they rearrange their DNA, generating a diversity of receptors that rivals the stars. Techniques like PCR, polymerase chain reaction, amplify minuscule fragments of DNA, allowing researchers to follow the fate of a single immune cell through the maze of an infection or an immune response. The tools of discovery, ever evolving, are not merely instruments of observation, but engines of imagination. With them, scientists ask questions once deemed impossible. How do vaccines teach the immune system to recognize threats it has never seen? What signals cause a T cell to awaken, to divide, to become a killer or a helper? How does the immune system remember, holding the shape of a virus in its mind for decades, ready to respond at a moment's notice? Some experiments are grand in scale, others almost whimsical in their simplicity. In the early days of allergy research, for example, Charles Richet and Paul Portier, in pursuit of the mechanisms of anaphylaxis, inject dogs with sea anemone toxin, only to find the animals react violently upon a second exposure. This paradoxical response, where the immune system, once sensitized, turns against the host with deadly force, reveals the double-edged nature of immunity, its capacity for both protection and harm. Other tales are more recent, yet no less remarkable. In the late 20th and early 21st centuries, the advent of imaging techniques such as two-photon microscopy allows scientists to watch immune cells moving through living tissue in real time. They see T cells patrolling the lymph nodes, B cells sprouting into antibody factories, dendritic cells capturing and presenting fragments of pathogen like heralds bearing urgent news. The once invisible war is now a living documentary, its protagonists caught in the very act of defense and decision. And yet, even as these tools grow ever more sophisticated, the immune system resists full conquest. Its rules are subtle, its alliances shifting. New discoveries of regulatory T cells that quell inflammation, of innate lymphoid cells that bridge the gap between immediate defense and adaptive memory, remind us that the battlefield is not static but alive, its contours redrawn with each experiment, each revelation. Tonight, as you settle deeper into quiet, consider the generations of scientists, some famous, many forgotten, whose hands crafted the tools and shaped the experiments that brought the immune system into focus. The microscope, the dye, the centrifuge, the antibody, the gene sequencer, each a key, each unlocking a new chamber within the labyrinth of immunity. Their stories are not finished, for the immune system, in all its complexity, continues to surprise and confound, to inspire both awe and humility. Beyond the horizon of our current knowledge, new questions beckon. What happens when the immune system falters, or when it turns its arsenal against the self? What hidden pathways remain, waiting for the next clever experiment or the next bold toolmaker? The battlefield is mapped, but never fully known. In the shifting twilight of discovery, we linger, half awake, awaiting the next dawn. Night has settled softly, and somewhere in the hush between waking and sleep, the story of immunity continues. Not as a recitation of facts, but as a meditation on what it means to be alive. Here, the immune system is no longer just a network of cells and molecules. It becomes an emblem, a metaphor, a window through which we glimpse the tangled complexities of selfhood and survival. In the quiet dark, as you drift on the edge of dreams, let us linger in the strange philosophical light that immunity casts across the human condition. Consider, if you will, the paradox at the heart of immunity. To defend the self, the immune system must know what the self is. It learns, from the earliest moments, to distinguish the familiar from the foreign, the proper from the intruder. Lymphocytes, those vigilant sentinels, patrol the body's boundaries and interiors, ever watchful. But this vigilance is not blind aggression. It is a careful discernment, a perpetual negotiation between the urge to protect and the imperative not to harm what belongs. At the core of this negotiation lies the doctrine of self and not self, a distinction that immunology has made famous. Each cell of your body wears a molecular badge, a unique set of markers crafted from your genetic code. Major histocompatibility complex molecules, MHC for those who prefer brevity, adorn the surfaces of your cells, broadcasting a biochemical signature of identity. To your cytotoxic T cells, these markers are the difference between neighbor and nemesis, kin and foe. Yet even this identity is not absolute. It is forged in the crucible of development, shaped by chance and experience. In the thymus, young T-cells undergo a kind of schooling. Those that cannot recognize the self are cast aside, for they would be useless. Those that recognize it too well are eliminated, lest they turn traitor and spark the fires of autoimmunity. What survives is a population of cells that knows you intimately, but not obsessively. They are tuned to the music of your body, alert for those notes that ring false. So, in a sense, your immune system is a memory palace of selfhood, a living archive of what it means to be you at the level of molecules and cells. And yet, paradoxically, this archive is porous. The boundaries it patrols are not iron walls, but shifting frontiers. With each breath, each meal, each brush of the skin, the outside world seeps in. Foreign proteins, harmless and harmful alike, are sampled, catalogued, sometimes tolerated, sometimes attacked. What then is the self that immunity protects? Is it a fortress or a river, a fixed identity or a ceaseless flow? In the immune system we see both. The body is a vessel of continuity, yes, but also an open system, forever in dialogue with its environment. The very act of defending the self requires a constant openness to what is not self. The immune system is not merely a wall, but 1,000 gates, opening and closing with exquisite sensitivity. This dance of openness and closure, of tolerance and defense, is not unique to the immune system. It echoes across the whole spectrum of life. It is the same dance that shapes the mind, the way we form identities, draw boundaries around the self, yet remain porous to others, to experiences, to change. Immunity, in this light, is not just a biological function, but a metaphor for existence. To live is to defend, but also to embrace, to remember, but also to forget. And here, perhaps, we glimpse one of the oldest human anxieties, the fear of the other, the longing for purity, the terror of invasion. Throughout history, disease has been cast as the enemy, the invader, the corrupter of bodies and societies. The language of immunity is steeped in metaphors of warfare, defense, attack, memory, vigilance. But the immune system is not a simple battlefield. It is also a site of negotiation, of compromise, of uneasy peace. The most dangerous immune reactions are not those that are too weak, but those that are too strong, too indiscriminate. Autoimmunity is the tragedy of mistaken identity, an immune system turned against the self it is meant to protect. In this, immunity mirrors the dilemmas of human society. The urge to defend the group, the nation, the culture can slip into xenophobia, paranoia, self-destruction. The immune system teaches us that survival depends not on absolute purity, but on flexible boundaries, wise discernment, and the capacity for tolerance. The healthiest immune system is not the most aggressive, but the most discerning, the most capable of distinguishing threat from harmlessness, danger from difference. Let us dwell for a moment on the phenomenon of immunological memory. Vaccines, one of humanity's greatest inventions, work by training the immune system to remember a foe it has never met. A weakened or inactivated pathogen is introduced. The immune system mounts a response, then files away the memory for the future. This act of remembering is not conscious, yet it is as profound as any cultural inheritance. The body learns from its history, building resilience not through isolation, but through encounter. There is something deeply human in this notion of embodied memory. Just as we learn from experience, so too does our immune system. Each infection, each exposure is a lesson inscribed in the tissues, a rehearsal for battles yet to come. Our bodies are palimpsests, layered with the traces of past encounters, resilient not because they are untouched, but because they have survived. And yet, memory is not infallible. Pathogens evolve, shifting their molecular disguises, slipping past our defenses. The influenza virus, master of mutation, outpaces our immune memory, forcing us to relearn its shape year after year. HIV, shrouded in a cloak of ever-changing proteins, evades recognition almost entirely. Here again, immunity mirrors the human condition, the necessity of vigilance, the pain of loss, the hope of adaptation. To be immune is never to be invulnerable. It is to be engaged in an endless contest with change itself. Consider also the phenomenon of transplantation, the attempt to bridge bodies, to share organs and tissues across the gulf of individual identity. The immune system resists this union, reading the foreign MHC molecules as a threat. Rejection follows unless the immune response is carefully subdued, the boundaries softened by medicine. In this way, immunity becomes a barrier to empathy, to the full embrace of the other, and yet with ingenuity and humility we find ways to coax tolerance, to persuade the immune system to accept what was once alien. The story of immunity is, in this sense, a story of coexistence, of uneasy truces and fragile alliances. The gut, that dense and labyrinthine organ, is home to trillions of microbes, a teeming metropolis of bacteria, viruses, fungi. The immune system must learn to tolerate these residents, to distinguish friend from foe not by origin, but by behavior. Here, the boundaries of self grow blurry. The microbiome becomes a part of us, shaping our digestion, our mood, even our immune responses. Who, then, are we, if so much of what makes us healthy is not human at all? This question lingers in the quiet, unsettling, and profound. The immune system, the very seat of our individuality, is also the means by which we are shaped by the world. Allergies, those overzealous reactions to harmless substances, remind us that immunity is not perfect. It is a system of judgments and errors, of risks and trade-offs. Asthma, eczema, hay fever, these are the price we pay for the flexibility and sensitivity of our defenses. The immune system, like the mind, is susceptible to phantoms, to imagined threats, to wounds that linger long after the danger has passed. In recent decades, immunology has turned inward, probing the links between mind and body, emotion and immunity. Chronic stress, it seems, can dampen immune function, leaving the body vulnerable. Grief, trauma, loneliness, these invisible wounds find echoes in the immune response. The ancient intuition that body and soul are entwined finds new resonance in the language of cytokines and neurotransmitters. To suffer is not only to feel pain, but to become more permeable to the world, more open to harm. Yet, there is grace here too. The immune system is not just a source of disease, but of healing. Inflammation, that much maligned process, is also the means by which wounds are closed, infections cleared, tissues renewed. The same cells that kill can also nurture, secreting growth factors, guiding regeneration. The balance between destruction and repair, attack and healing, is delicate, always shifting, always at risk of tipping too far. But it is this balance that makes recovery possible, that allows life to persist in the face of injury and loss. As you rest there, listening, perhaps you feel the pulse of your own body, the slow rhythm of breath, the quiet thrum of blood. Within you, a trillion tiny dramas unfold, unseen and unceasing. Cells die and are reborn. Antibodies drift through your veins, silent sentinels. Microbes converse with the cells that host them, exchanging signals in a language older than words. The immune system is not an army, but an orchestra, its harmonies ever-changing. And what of the future, you might wonder? What new alliances and adversaries await our immune systems in the years to come? With every advance in medicine, we deepen our understanding, but also our responsibility. Immune therapies, engineered antibodies, designer T-cells, personalized vaccines, hold the promise of healing once incurable diseases. Yet they also raise questions about identity, autonomy, and the limits of intervention. If we can reprogram the immune system, can we also reshape the self? If we can silence autoimmunity, can we also silence the parts of ourselves that cause pain, but also growth? There is a humility in the face of such questions, a sense that immunity will always exceed our grasp. For all our knowledge, the immune system remains a wilderness full of surprises, dangers, and delights. It is a system built not for perfection, but for survival, for the messy, contingent, ever-changing realities of life. To be immune is not to be untouched by the world, but to be shaped by it, marked by every encounter, every wound, every act of healing. In this way, the story of immunity is the story of humanity, a tale of boundaries and crossings, of memory and forgetfulness, of vulnerability and resilience. We are not isolated fortresses, but open systems, forever negotiating the terms of our existence. Our immune systems, like our minds, are shaped by what we encounter, by what we love, by what we survive. And so, as the night deepens and your thoughts grow gentle, let yourself drift on the currents of this immortal dance. The immune system is not a guardian standing apart, but a part of the self, a companion on the journey, ever vigilant, ever changing. In its ceaseless work, we glimpse not only the mechanics of life, but the mystery of being, fragile, resilient, open to change, forever unfinished. As our journey through the wondrous realm of the immune system drifts to a close, we float gently like a comet tail, fading into the hush of space, marveling at the quiet battles waged within us that keep us safe and whole. May this peaceful reflection settle softly in your mind, inviting you to return soon for more Dreamtime tales, where the mysteries of life twinkle like distant stars, waiting to be explored.